Did you know a virus can hide from your immune system like a master spy? That's what HIV does. Today, I will discuss HIV. HIV, or Human Immunodeficiency Virus, is one of the most complex and deadly viruses known to humanity. Unlike other viruses, HIV specifically targets the immune system, the very system that protect our body from infections. It mainly attacks CD4 plus T-cells, which are essential for immune defense, gradually weakening the body and leading to AIDS, means acquired immunodeficiency syndrome, if left untreated. In these sessions, we will explore how HIV spreads and replicates, why it's so hard to develop a vaccine, the structure of the virus and its genetic makeup, how it tricks the immune system, the global burden of the disease today. Let's begin by understanding its structure, genetic behavior, and how it interacts with the human cell. In 2024, approximately 40.8 million people were living with HIV globally. According to Joint United Nations Program on HIV/AIDS, means UNAIDS, approximately 6.3 lakhs people die every year due to AIDS as of 2024, and near about 1.3 million new cases every year. United Nations Program on HIV/AIDS, means UNAIDS, reports that 65% of people living with HIV in 2024, approximately 25.9 million, reside in sub-Saharan Africa. Eastern and Southern Africa account for 20.8 million, and Western and Central Africa for 5.81 million. The Asia and Pacific region accounts for about 6.7 million people living with HIV, which is roughly 16.4% of the global total. The remaining regions, such as Western and Central Europe, North America, Latin America, Eastern Europe, Central Asia, Middle East, and North Africa, collectively account for about 18.6%, approximately 7.6 million. According to the National AIDS Control Organizations, or NACO, and UNAIDS, an estimated 2.54 million people were living with HIV in India in 2023, with an adult prevalence rate of approximately 0.2%. This number reflects a slight increase from 2.47 million in 2022, indicating a slow rise in recent years. The question is, why cannot we make a fixed vaccine against the HIV viruses? Answer is that we cannot make a fixed vaccine against this virus because HIV's GP120 protein is a really tricky molecule. The gene that produces GP120 protein undergoes rapid alterations and mutations, leading to high genetic variability due to the error-prone reverse transcriptase enzyme during viral replication. As a result, there are many antigenic variants of GP120 protein. This constant change means our immune systems cannot produce a single fixed antibody against it. The virus keeps changing its face. Not only does GP120 have this conserved, means less mutable region, but it also makes it harder for antibodies to bind effectively. Then scientists are working on a vaccine targeting stable part like GP41. HIV resides within a protective bilayer envelope, allowing it to fuse with the host cell membrane and enter the cell. Once inside, the virus hijacks the host machinery to replicate its genetic material. Newly formed viral particles are then bud out from the host cell, cloaked in a portions of the host own membrane. In this way, HIV effectively steals the host cell covering to disguise itself and evade immune detection. There are two types of HIV are found in the world, HIV-1 and HIV-2. HIV-1 is the most common type, found all over the world, whereas HIV-2 is mostly limited to the West Africa. Formerly, HIV was known by different names such as the Human T-cell lymphocytes virus, HTLV3, lymphadenopathy associated virus, LAV, and AIDS associated retrovirus, ARV. A man who becomes HIV positive produces more than a billion virus every day. HIV is a very dangerous virus that is spherical in shape. HIV is basically an RNA retrovirus. In this virus, a process called reverse transcription takes place. The word retro means backward, which is why HIV is called a retrovirus. The enzyme reverse transcriptase helps in the process of reverse transcription. So, let's explore some internal and external aspect of HIV. To begin with, HIV is an enveloped virus. The envelope is a lipid bilayer surrounding the viral matrix, giving the virus its spherical shape. This envelope is derived from the host cell membrane during the budding process. Please see this video. Beneath the envelope, there is an icosahedral cell called the matrix protein P17. The core consists of the cylindrical capsid protein P24, which surround the viral genome. Let's discuss the HIV genome and its gene. HIV is a single-stranded RNA, ssRNA virus. Its genome consists of two identical copies of the positive sense ssRNA. These RNA strands are associated with specific protein and are linked at their 5' ends. The genome of HIV consists of 9 genes in total: 3 structural genes, ENV, GAG, and POL, and 6 regulatory genes or non-structural genes. They are TAT, REV, NEF, VIF, VPR, and VPU in HIV-1, and VPX reflects VPU in HIV-2. Let's learn about the enzyme and other properties of the HIV. HIV belongs to the retroviridae family, possesses several unique enzymes that facilitate its replication inside the host cell. One key enzyme is reverse transcriptase, means RNA-dependent DNA polymerase, which convert the viral RNA into complementary DNA, means cDNA. Integrase then helps insert this viral DNA into the host genome, while protease cleaves viral polyprotein into functional unit, essential for maturation of the new viruses. The enzyme ligase may also assist in joining nucleic acid fragments during integration. HIV shows oncogenic potential in certain contexts and undergoes a high rate of mutation, particularly in genes like GP120, making vaccine development challenging. Additionally, it is species specific, infecting mainly human CD4 T-cell, and is characterized by its classification under the retrovirus family due to its reverse transcription mechanism. HIV resides within a protective bilayer envelope, which allows it to fuse with the host cell membrane and enter the cell. Once inside, the virus hijacks the host cellular machinery to replicate its genetic material. The question is, how does this happen? Let's explore. GP41 means glycoprotein 41 is a protein that facilitates the ultimate fusion of the viral lipid membrane with the host cell lipid membrane, a deadly fusion that allows the virus to enter the cell. The HIV virus membrane fuses with the membrane of T cell and macrophage during infection. GP120 is another viral glycoprotein that helps the virus attach to the target cell by binding to its surface receptor. HIV has a strong preference for infecting CD4 positive cells. CD4 positive cells includes T helper cells, monocytes circulating in the blood, macrophages, microglia in the brain, specialized dendritic cell under the skin, Langerhans cell, follicular dendritic cell in lymph node. First, GP120 binds to the CD4 receptor on the host cell. This triggers a conformational changes in GP120, revealing another arm or domain that interact with co-receptor. Here is the picture. This co-receptor is actually a receptor for chemokine. In the case of macrophages, the co-receptor is CCR5, means chemokine receptor 5. These cells don't express CXCR4 as a co-receptor. In the early stage of infection, HIV primarily uses the CCR5 co-receptor to enter and infect CD4 plus T cells. CCR5 is mainly found on the surface of the T helper cell and macrophages. CXCR4 is another chemokine co-receptor used by certain strain of HIV during later stage of infection or different host cell. Some people have natural immunity to HIV infection due to a mutation in the CCR5 gene. These individuals may test positive for HIV but don't develop AIDS. This mutation can be homozygous, means both maternal and paternal allele mutated. This provides strong protection, and heterozygous, only one mutated allele, provide partial resistance. Interestingly, people of Northern Europe descent, particularly white individuals, are known to carry this CCR5 mutant gene. Approximately 1% are homozygous and around 10 to 15% are heterozygous. On average, after HIV infection, it takes 7 to 8 years for AIDS to develop in untreated individuals. Now let's learn about the replication cycle of the HIV in human cell. HIV is like a hacker that sneaks into a cell, rewrites its code, and makes the cell produce more viruses. A man who becomes HIV positive produces more than 1 billion viruses every day. Once the HIV virus enters the host cell, our cellular protease act on the capsid protein P24. This protease helps disassemble the capsid releasing the viral RNA into the cytoplasm of the host cell. At this stage, the enzyme reverse transcriptase carried by the virus begin to infection. Reverse transcriptase has two crucial enzymatic activity. One, RNA dependent DNA polymerase activity. It synthesizes a complementary DNA strand using the viral RNA as a template. And number two, RNase age activity. Here age means a hybrid. It degrades the original RNA strand of the RNA DNA hybrid. Following this, the enzyme synthesizes a second DNA strand, resulting in a double-stranded viral DNA. This double-stranded DNA is then transported into the host cell nucleus, where it becomes integrated into the host genome. This integrated form of viral DNA is called proviral DNA. It can insert itself at multiple sites within the host DNA, and once integrated, it becomes a permanent part of the host genome. Come, let us clarify our understanding of a few terms. The ENV gene is responsible for producing the envelope glycoprotein of HIV. The capsid is icosahedral in structure, meaning it has 20 faces. GP41, glycoprotein 41, is relatively stable and does not undergo frequent antigenic variation. In contrast, GP120 is highly unstable and prone to antigenic changes, allowing HIV to evade the host immune response. The HIV genome contains two identical RNA strands, not complementary to each other. These are called the dimer. Both RNA strands are of positive polarity, means positive sense RNA. A positive sense RNA, like typical messenger RNA or mRNA, has a 5' cap and 3' poly A tail, and it can be directly translated into protein by host ribosome. In contrast, negative sense RNA is complementary to the positive sense RNA. It cannot directly translate it into protein and usually lack both a cap and a poly A tail. It must first be transcribed into positive sense RNA by an RNA dependent DNA polymerase. In normal cellular transcription, a DNA dependent RNA polymerase transcribes RNA from a DNA template. However, in the case of the HIV, an RNA dependent DNA polymerase, also known as the reverse transcriptase, is used. These enzymes synthesizes DNA from an RNA template, a process known as the reverse transcription. HIV remains a formidable global health challenge due to its complex structure, a rapid mutation rate, and ability to evade the immune system. Its targeting of CD4 plus T-cells facilitated by glycoprotein like GP120 and GP41 undermines the body's defense, leading to AIDS if untreated. The virus, high genetic variability driven by the error-prone reverse transcriptase enzyme make vaccine development exceptionally difficult. Despite advancements in treatment and prevention, the global burden, particularly in sub-Saharan Africa and regions like India, underscores the need of the continued research, awareness, and equitable access to care to combat this persistent pandemic. Thank you.